confused on the first one because
Okay.
I was not sure about the first question.
Okay.
Uh, yeah, so I don't know how to analyze the question to find out which one is the right sample there.
I see. How do you feel about all of this up here overall?
Overall, like I understand that the A sample A has the longer segment and the shorter segment, so
Hmm.
it is a combination of like I mean it would be around a 4,500 plus 500, so the total circumference is 500 base 5,000 base pair.
Yes.
Uh, I see that, but I don't know if I answered question one right. I haven't seen your email yet. Sorry. So.
Oh no, that's
But
perfect. Yeah, yeah. That's the only thing that I included in my email was that your question one answer was off.
uh-huh,
So looking at this question, obviously you want to verify the success of this like ligation experiment.
uh-huh,
Um, One way to verify that the ligation experiment worked is that you could like cut up this recombinant DNA. So hypothetically, when you have this 4,500 base pair insert plus the 500 base pair, or the plasmid plus the 500 base pair insert, yeah, the whole thing will be 5 kilobases, which is what you see in sample B, right?
yes, okay, 5,000, yes, okay,
Yeah, but to validate, so there's technically two ways of validating that you have the right product. You could either run it uncut, so like completely intact here, which is sample B would be the answer.
uh-huh,
Which I mean, spoiler alert, that is the answer that we have on our key. But looking at sample A, another way to validate is actually cut it with these restriction enzymes. That's kind of like what the field uses. So
uh-huh,
looking at sample A, after you cut it with KPN1 and Beagle2, which are just the two restriction enzymes used here, you do have the correct bands. So in my opinion, both samples A and B verify that you have the right plasmid, but the key specifically like wants this one.
so but how do we differentiate? So like does it mean that in A the two parts were not the DNA ligase was not used properly, that's why they were not together? Is that what it means?
Okay, that could be another interpretation actually. Is that yeah, in sample A, maybe you combine both the plasmid DNA and the insert DNA, and there was no ligating happening. So
Okay, so
yeah,
but answer for question one could be A or B, but for this it's B, is that what it is?
yes, yeah. Question one, I guess the more correct answer would be sample B because yeah, because that first sample or that first lane for sample A could just be both DNA fragments unligated.
Okay, so what about question two then? Observation of sample C. So sample C is off too, right? So the ligation step was missed or it failed. Will that work?
Correct. Yeah, because this is not the like looking at the other three answers also, it's not the expected recombinant DNA.
Because there is I mean, we are short of 500 base pairs there.
Exactly. Yeah, so sample C, you just have your plasmid DNA there and no insert, which usually indicates that your ligation step was wrong.
Okay,
Um, yeah, for
okay,
some reason your insert just isn't there.
okay. So yeah,
Yeah. Yeah, but question two is just long story short, the ligation step failed.
fail. Okay, awesome. I think I'm good. Let me see. So everything else is good, right?
Yes, yeah. Everything else looked good. How do you feel about this? I feel like the other thing that was introduced in this recitation that was a big one was this
Blue-white
black,
screen.
yeah, like the colony screening. Did this
Yes.
make sense?
Yes. So the blue colonies mean that the laxine was intact and functional in the sense that they were able to produce the beta galactosidase, right?
Yes, that's right.
Okay. So yes. So when you have the blue colonies, that means the lactose gene was present, but it's still the plasmid took the insert, right? They took the non-recombinant ones, but they were able to produce LAC-Z gene.
Correct, correct. Yeah, the blue colonies have the intact LAC-Z gene, which means that the gene of interest isn't there, like it wasn't inserted into the plasmid, but the bacteria still took up recombinant DNA.
Okay. Okay. Yeah. Yes. It looks interesting. I wish I was there to do the lab, but I'm in the Eastern Zone.
Definitely, yeah. I mean, this is a little, I mean, I think it's a little bit of an outdated protocol to use this, but it's still like a great screening method.
Awesome. Awesome.
Yeah. Just because when you have like so many colonies on a plate and, ligations aren't very foolproof.
Okay.
So sometimes you just get weird colonies on the plate and you don't know why. Sometimes, yeah, this is a great reporter.
Uh,
Yeah.
okay. So pretty much first and second there was a lot of math, which was fun, but this one is a lot of theory. Too much to
Definitely.
too much to remember.
Yeah.
Uh, I'll do my best. But thank you so much, Luke, for all your help. Thank you.
Yeah. Yeah, of course. I'm glad that I could help.
Yeah. Have a great day, and thank you so much.
Yeah. Yeah, of course. Best of luck in future courses too.
Thank you. Thank you. Bye.
Bye. All right. So while I have the recording going, I could just go over the questions. Just for people who weren't able to make the office hour. So we kind of just went over questions 1 and 2. One thing that I wanted to mention is that don't worry too much about these restriction enzyme names. They're just two enzymes that are frequently used um, for cutting up DNA. Obviously, like all of these restriction enzymes, Mario might have mentioned this in the video, were found in different bacteria and archaea. Obviously, they need, certain defense mechanisms against viral DNA, because these viruses can still infect these prokaryotic cells. So what people did is that they cloned some of these enzymes out of the bacteria that cut up viral DNA, and they found that they, cut at specific sequences. So, there's hundreds of these things out there, and they're just cool tools to kind of um, have like a lot of flexibility when it comes to creating these clones, especially when you want to use a ligation step that requires specific restriction enzymes for plasmid and insert DNA. Um, so yeah, we kind of went over samples 1 and 2. Here you're looking at gel electrophoresis again, which is very important for the cloning workflow. I also highly recommend that you guys just kind of understand the cloning workflow as a whole, starting from, doing a PCR on a genome to get, this insert DNA from some genome of interest, and then understanding what a plasmid is overall. There's a lot of good videos that I think Tia Mario included on, or just below his video that I also recommend you guys watch. So then looking at sample C, obviously what you'll see here is that the ligation step just didn't work out. You just have the plasmid DNA on its own. So question 3 kind of takes a step back and looks at gel electrophoresis overall again, which we've already kind of gone over. I think it was in recitation 8, which sample would likely travel the furthest in the gel. You'll see that, obviously the smaller DNA fragments travel the furthest because, I think of it as an obstacle course where you have all of these little agarose beads in the way, and the DNA is trying to get down the gel. So obviously, smaller DNA fragments will have an easier time getting through like the cracks and crevices of the agarose gel. So then question 4 asks which experimental setup would best confirm that a PCR product is the correct insert for cloning. So here's where we talk about controls, because when you're running a gel and you have a PCR product, obviously the DNA ladder, which you always run in a DNA gel, the ladder is always going to tell you how big the PCR fragment is, but you're always going to want another sample that has a known size and around the same size that you're looking for with the PCR product. So out of these 4, you'll see that one of them talks about, positive controls. And then question five, which step ensures that only bacteria that have taken up a plasmid can survive on selective media? Now, this question kind of gives you the answer because the selective part usually pertains to some sort of, resistance gene. So when you include antibiotics on transformation plates, it's going to kill every bacteria except for the ones that contain a resistance gene. So usually when you engineer these bacterial clones, you'll include some sort of here, let's see here. You'll include some sort of like resistance gene. So for example, let's pretend that this is our plasmid of interest. Great art skills. So here, right, you're gonna have like some gene of interest, but um, the rest of the vector, like pretty much everything you see around the gene that's part of the plasmid, is called the backbone of the plasmid. So something cool that you can have, let me see if I can change this here. Something that you can have in the backbone of the plasmid is another gene. Let's pretend like there's something here, and this is present in all plasmids. Let's pretend that you're using ampicillin on like your selective plate. So the plasmid itself can have this ampicillin resistance gene. Yeah, it's hard. So that way, any like bacteria that has this plasmid can also have this resistance gene and be able to survive on the selective media. So I hope that clears things up on why we use that. Because obviously you can have millions of bacteria grow on the same plate if you're not careful and have some sort of selective marker. So bacteria can, take up this plasmid DNA that has the ampicillin resistance gene or whatever. A lot of people use kanamycin. Some people use chloramphenicol. There's just a lot of different antibiotics that people use, but a lot of bacteria will take up your um, plasmid of interest, but sometimes the plasmid won't have the actual gene of interest just because a lot of cloning experiments, such as, ligations, aren't very efficient. So they use this LAC-Z reporter gene, which if you look that up on Google or watch the video about it, there's a lot of good information about that as well. So then question seven talks about or asks why must restriction enzymes be chosen based on both the plasmid and insert sequences? Once again, you're looking at, how the restriction digest can lead to a successful ligation, and obviously you need compatible ends, right? That are present from the restriction digest. Question eight, in the cloning workflow, what is the most direct purpose for using alkaline lysis after colony selection? So lysis, the product of that is a bunch of, lysed bacteria, which means that the bacteria are pretty much broken open. So you have this like almost bacterial soup of the junk from the bacteria that you don't really want, proteins, DNA, and RNA. So then you use this protocol that then separates the DNA of interest from the rest of the bacterial guts, pretty much. Question nine asks, a gel shows DNA bands in the negative control lane after PCR. What should the student conclude? You never want a band in your negative control. That's why they call it a negative. So if you see something here, that means something else got, contaminated or just really got in your PCR experiment that you don't want. So then question 10, a student cuts a plasma with EcoR1 and their insert with HIND3. These are two other restriction enzymes that are widely used. After ligation, no colonies grow. What likely went wrong? So when I was talking about the enzymes earlier, you'll see that there are these 6 nucleotide sequences that these enzymes cut around. So for EcoR1 specifically, it cuts at GAA, TTC, going 5' to 3'. Then HIN3 is AA, GCTT. So what happens here is that you'll have these sticky ends where I guess you could call it the coding strand, is going to have the sequence G, and then, it'll have like an overhang here. So that's what the sticky end means. So what you're going to notice is that when you cut um, one plasmid or one, yeah, so the plasmid's cut with equal R1, and then the insert with Hin3, you're going to see that the ends are not compatible. And so that will probably result in an unsuccessful ligation. So that's why you usually cut the plasmid and the insert with the same restriction enzyme. So question 11, which of the following are real world uses of recombinant DNA technology for producing medically relevant products? So this is why we care about, bacterial cloning. So you'll see that there are a few options here that are correct, and are cool applications for actually creating these or for actually using this technology, right? And then finally, question 12, to confirm that you have obtained the correct recombinant DNA, which of the following experiments are appropriate to perform? This kind of I guess, links back to the first question, in validating that you have the correct option. So definitely refer to that one. Um, Refer to that question to see what it is. But once again, let me know if you have any questions about this recitation or anything leading up to your exam 3 and final. But yeah, best of luck.